Now I'd like to introduce Dr. Lori Leclaire, who will introduce our keynote speaker. Thank you.
Thanks. So welcome everyone on this beautiful fall Saturday morning. It's a great pleasure for me to welcome Dr. Beall to the University of Vermont Medical Center. Dr. Beall started the early part of his career at the Cleveland Clinic, and then in the 1980s moved to the CF Foundation, where he remained until his retirement late last year. Initially, he started out as the Vice President of Medical Affairs, but in the 90s became the President and CEO. It's under his leadership that the CF Foundation became widely recognized and known as the real model of what you can accomplish when you leverage the best of academia with the best of industry. And he is responsible, really, for many of the therapeutics that are currently available to our patients today. Importantly among them is ivacaftor and Orkambi. And while he's certainly very well known for that, I think he is almost better known within the CF community of CF providers and care team members as a staunch supporter of us. And really, the CF Foundation has established what it means to provide interprofessional, patient-centered care that now really the rest of medicine is clamoring to follow. So I'll end with that and welcome you to the podium.
Thanks, Lori. Can you hear me? I assume this is on. Got to be careful when these things are on or off these days, you know. We might get into real trouble, you know. And my wife is back here, Mimi, so I got to be really careful what I say. And I understand this is being recorded, but so the mic is on. The mic is on. So anyway, thank you, Lori. Lori and I know each other not only from her being head of the adult program here, but she was on our center committee for a number of years, and we had a lot of opportunities to talk because my family, my son lives down near Montpelier, and so we talked about my love of Vermont, Mimi's of my love of Vermont, the culture, the people, and everything, and the beauty, obviously today, of what this, and the people. God love the people. We've had an opportunity recently to learn the importance of the people and how they rally around each other, and which is great. So anyway, I really appreciate the opportunity to be here. As Lori said, I stepped down as president and CEO last October now, and but people said, "Hey Bob, don't jump off the train. Don't get off the train. Don't get off the train." You know, and they said, "What would you like to do? You got to do something." And I said, you know what I miss most about stepping down and having been the CEO and having to worry about all the business stuff and all the stuff you have to work with when you're dealing with 600 employees and so forth, was the opportunity to go out and meet the people and meet the parents and meet the grandparents and the aunts and uncles and friends in this amazing community. Because when I first got to the foundation, you know, back in 1980, we were so small, we only had, we did everything by a support service contract. And so I would have to almost answer the phone, and a lot of times I would hear mothers call me up and they say, "Oh, my child is being sweat tested tomorrow. It's going to be sweat tested tomorrow." And I picked up the encyclopedia, and that dates me, okay? Encyclopedia, okay? It picks up the encyclopedia and addiction, and they talked about, you know, this is, you know, a disease still where the patients weren't living into the teens, and you know it's a death sentence to people with cystic fibrosis. And I'd have to say, throw away the dictionary, throw away the encyclopedia, because this is very different today than most likely when your encyclopedia was written, and the misperceptions. But anyway, so I said I wanted to go out and meet the patients, and so they said, "Well, what are you going to talk about?" You know, and I said, what I really want to talk about is, can we do better? Lori just mentioned about Orkambi and Kalydeco, and I heard about some great success stories with Orkambi and some great success stories with Kalydeco, but the question is, that's not enough. And so it really leads back, can we do better? And because everybody is excited about, for the first time, we have over half of our patients are over 18 years of age. There was a time we didn't have adult care centers. We only had pediatric centers. We had to fight to get a good adult care centers. Median life expectancy is 41. It's still in the vicinity. But all of these things have helped to make a difference. And so, but people say, can we do better? Well, obviously, I'm here to say, yes, we can. And what I'd like to do is spend a few minutes to speak to you about, why I am so confident and that we will and are going to do better. And one of the things that I have learned over the last year, is this too much trouble if I walk?
No, do your thing.
Can I do my thing?
Absolutely.
Oh, okay. Well, you know I'm going to walk. Anyway, so one of the things that we really have that we need to be proud of, and there's no other disease like it, and it's evidenced by just the participation here this morning. We have people from industry here, from Genentech here, and I'm sure there are others that I might miss. We have people from patients, we have uncles, we have grandparents, we have our physician community, we have medical students here. This is a community. There is nothing like this in any chronic disease. A lot of the chronic disease, they're fighting against each other, 2 or 3 different organizations, but this is a community. And it's that community that I think is so important for us to be able to achieve what we want to achieve with our mission. So, and I need to go to move here to the next slide here. So, whoop. Okay. So this is, the big word now is ecosystem and all that kind of stuff. So this is our ecosystem of cystic fibrosis. But it really started out with the foundation in 1955. When a group of parents, and this is for some of you younger folks in the audience who maybe have never heard the story, but they started out with a group of parents who were totally frustrated by the fact that they were told that their children were not going to live to school age. Doris Tolson and Milton Graub and a few parents all got together and said, we got to do something about that. 1958, and this group really together, they created something called HOPE, when there was no hope. And one of the things I think about hope is a very kind of an important amorphous glue that has been able to be passed on for the last 60 years amongst these families and amongst the people and amongst this amazing community. And that glue bonds us, the bonds this community to something that no other organization has. And it provides strength when things go up and down with this disease. And there have been good times, bad times, disappointments, because we're dealing with a chronic disease. And it's that sense of hope that has been carrying us through. But there's another important thing that community really initiated very early on, was the concept of innovation. We're not going to do things like everybody else did. In 1965, they started the Care Center Network. They started out with 4 centers, mostly in New England. And now we have 113 pediatric centers, but we almost have the same number of adult centers. That was unheard of. They provided minimal support to these groups, but one of the things they started to do was to collect data with our patient registry. You all have signed consent forms for your children or yourselves to have data collected. And we have collected that data since 1965, and we have a natural history of this disease. And when the FDA approved Kalydeco, I was with Janet Woodcock, who was the head of the FDA at that point, drug division still is, and she says, Bob, the most important thing that the CF Foundation brought to the effort was the natural history of this disease. This was in 1965 that these people with very little money started. And it's that innovation that has carried this organization forward. In 1980, we set up our own research centers. At that point, cancer centers were being established, arthritis centers were being established, but CF had no science. So what did we do? We said, we're going to set up some centers. And we're going to bring people from the clinical side, with basic scientists, with microbiology, and all of that, and bring them all together in institutions around the country. We had no money. We had no money. And a wonderful woman called Mary Weiss, called me on New Year's Day in 1981. She said, "Pop, I just got our first $1 million." $1 million for the CF Foundation then was a heck of amount of money. I didn't say hell, okay? I'm doing good, okay? Okay. I'll be happy. Amazing, just amazing. Anyway, I'll be careful. And the most important thing was we've had the parents support the research. And those research centers, we have 13 of them, became a focal point for our efforts. And then in 1989, what did we do? We went out and found the gene for cystic fibrosis. And we did it the old-fashioned way. We paid for it ourselves. We didn't rely on the government through the Human Genome Project to do it, because they were 12 years after we found our gene. So we did it the old-fashioned way. And so that's innovation. And then we'll come back to this in a couple minutes. In 1998, 1999, we couldn't get industry involved in cystic fibrosis. And we'll go back to that in a couple minutes. But, we started our therapeutics development program. But inherent in our therapeutics development program was a clinical trial network. Because no company is going to get involved in cystic fibrosis. No company is going to get involved in cystic fibrosis if they can't have access to the patients. So we started the clinical trial network, of which you all are participating in that, where you have a research coordinator here provided by the CF Foundation. But the most important thing about that is we started to support industry, and we've created this community. So this is, I don't like I hate PowerPoints, by the way. I only got 5, okay? And I probably will screw them up, okay? Cystic Fibrosis Foundation first worked with academics, creating this first part of what we do with research, care, training. The CF Foundation supports and trains many of the physicians that take care of CF patients. Don't know if you knew that. We have some of the most active pediatric training program and adult training programs. And then what I said here is we bought our biopharmaceutical partners, and I'm going to talk about that in a second. So that is the ecosystem. That is a community. And it's reflected right here. We have people here from Genentech. One of the major biopharmaceutical partners. We have Vertex. We have others, I'm sure, that are here. But we've created a community. And those scientists that are working in the laboratory, whether it's at Gilead or Genentech or Vertex, are as passionate and as committed as each and every one of you are to making a difference. And that is something that really no other organization has. Because, we give $56 million a year to Pfizer, to develop new drugs. And so there is a community that exists. The other reason, so the first reason I really think we're going to be successful is this passionate community, as glued together and brought together by the concept of hope. The second thing is where we are today in this disease is based on good science. Good science. How many times have you looked at NBC at night and they show some sort of a study that is taking place and they talk about this could be a breakthrough and you never hear about it again? Never hear about it again. That has not been the case in cystic fibrosis. We have been above the fold on newspapers, we've been on TV, and so forth. All of our work is based upon good science. And that science really goes back to the early 80s with our first research centers that we put into place, where a group of scientists were putting electrodes in the nasal passages of CF patients. They found that there was something electrically different, and they developed a hypothesis. Now, you will know from talking to your children that a hypothesis is critical to scientific method. And they learned and they said, what's wrong is chloride doesn't get out of the cells properly in airway patients. When chloride can't get out of the airways, it draws in water from the airways. You get these very, very thick, tenacious secretions. The bacteria live there, and you set up this cascade that we know as cystic fibrosis. So that was a fundamental hypothesis. And that hypothesis still stands today. In a few minutes, when I'm talking about looking at trying to find one-time cures for cystic fibrosis, it is still based upon that hypothesis. How can we get chloride out of the cells? If we were chloride molecules, how are we going to get out of that door? How are we going to get out of that door? And that is so fundamental. It has stood the test of time for 30 years. And I tell you, there's not many hypotheses that stand the test of time for 30 years. But it has given us the opportunity for progress. And that's what we found when the CF gene was found, because we didn't know why chloride couldn't get out of the cell. We didn't know why it couldn't get out. But, when we found the CF gene, we found that the gene made a protein. And that protein was the channel that allowed chloride to exit the cell. So think of that door right there, and we all want to get out of there, as being defective in cystic fibrosis. And what we really want to do is to find ways to open up that channel, and that channel actually is the protein all around that door frame that allows things to move out the door. CFTR is the protein that is defective. So early 1990s, we worked on gene therapy. We started about gene therapy. Disappointing, but you have some work going on right here now, still in gene therapy, that I think is very, very important. But the other thing we decided to try to do was, could we find small chemical compounds, small molecules, oral drugs is what we would say, that might be able to take that defective protein that's in that door and correct it, open it up. And so we had this idea that and chemical compounds, go over the chemistry lab. They probably have 200 compounds over there. But go to certain libraries around the world, and they may have 200,000 that you can buy. And all of that, you can buy these libraries. And we were trying to randomly screen these chemical compounds to see if they could, am I out of focus?
You're good.
Okay, thanks. See if they could open up that channel. I'm sorry I have to move around. But anyway, to open up that channel. But the academic community, it would take them about 2 or 3 days to screen 2 or 3 compounds. And how do you go through a library of 200,000 or 400,000 when it takes all that time to do that? When you're in academics, you've got a lot of committee meetings, and you got to go to committee meetings and dealing with issues that are inversely proportional to inversely important to the time spent on them. So they were kind of slow in getting there. And I see the academics over here laughing. You can see them over in the corner. Yeah. So anyway, the most important thing was, we said, we've got to get industry to help us. And we read where industry could screen maybe 1,000 compounds a day, 200,000 compounds a day, or 20,000 compounds a day. So I called up 8 companies that were doing this. Only 2 returned my phone call. I think the other 6 thought I was trying to raise money for the Cystic Fibrosis Foundation, never returned my phone call. But 2 returned our phone call. And that was one, Aurora Biosciences. And Aurora was a small biotech company, and the other was Genzyme, which is still working on a lot of areas in cystic fibrosis. And so what they did is they applied high throughput screening to see if we could find 1 or 2 compounds that might be able to open up that defective door. And the chances of finding that were really, really very remote. We didn't know if we were even going to find any that could do that. I often said I was 6'3", like Preston Campbell is, or 6'5", and a full head of hair when we started that, because, we didn't know. And we invested $4 million, then $40 million, then $75 million to see if we could find any drug that could open up that channel. But it's all based upon great science. And so that led to what we now know as the first drug that was approved in 2012, Kalydeco, for a group of patients with a certain mutation, because the way they studied it, they had cells that were derived from different patients who had undergone lung transplants, and they got the cells, they grew the cells, and they tested chemical compounds on one of the cell lines called the G551D, which is in about 3% of our patients, has a certain mutation, and they found a drug that could open it up. Now, that was for 3% of our patients. So I can remember them asking us, are you going to have problems from your community if we only start working on a drug that works for 3% of our patients? And I said, nope, because this is important proof of concept. If we can get this cross the finish line first and open up a proof of concept for our patients, this will be incredibly important. But at the same time, we were looking for the most common mutation, the Delta 508 patients, because that's a little bit more complex than just opening up the door, just opening up and correcting the CFTR. In the Delta 508, you got to move the drug from, or the protein from inside the cell over to that door, and then you got to open it up once you get there. So you need two drugs. And that was the basis of our Comi that was approved last year. And that is for the Delta 508 patients. And now it's for the Delta 508 patients with 2 copies of the Delta 508. So they have 2 copies. Then there are other patients with 1 copy of Delta 508 and other, which they're heterozygous for those alleles. We don't have a drug that works for that group of patients yet. So that's where we are today. But it's all based on great science. And it's making a difference. One of the differences that I tell patients or individuals with cystic fibrosis so often, that it's just not the improvement in lung function that you see. The most important thing is that in every single patient, there's a natural rate of decline over time. And with Kalydeco, that rate of reduction is almost reduced by about 80%. So if you look at that, that extrapolates to extending life in these patients. And I think that is the most important thing, because we're treating the basic defect. Up to this point, we've always treated the symptoms of cystic fibrosis. Now we're treating the basic defect in CF. So that's where we are today. That is where we are today. We have an amazing community where we want to have committed physicians, committed researchers. We have families that are passionate about making a difference in the lives of their children. We have great people who want to work with us. We work with on the Hill. We are recognized for who we are. And then we have great science. So that really brings me to this point of, that's where we are. Where are we going? Because I think that's what is so critical to where we are. And I think that's the next point, hopefully, of a PowerPoint that I might have here. Oh, there's one other thing. Along the way, I had talked about the development of drugs to treat the basic defect. But, one of the things, and Marlene back here is going to love this next slide, because, we're going the wrong way. I don't want to love that slide. There is no health without mental health. Marlene, I did that just for you, okay? Marlene and my grandchildren, my grandson have played, her son and my grandson played soccer together for years. So we know her from the sidelines here in Vermont. And, but we've been innovative, not just with drugs and our care center, but now we're dealing and have the opportunity to bring other factors into our program. And we've just started a new mental health initiative. And you all have a new person, Lori, providing the opportunity for more mental health people going to be screened, pediatric and adult, because we found out depression in these people with cystic fibrosis is higher. So it's not just the pills we're worried about. We're dealing with the whole person, and I think that's really critical for us to think about. So anyway, we got that one in. Okay, Marlene? All right. We have to say now probably the kids would be on opposing teams. So anyway, so where are we going? Where are we going at this point with taking care of the patient? And this slide is probably the only PowerPoint slide you'll ever see where we're going backwards. We're going to go from right to left, because over here is where we would love to eventually end up, is with a one-time cure for cystic fibrosis. So right now, I want us to focus on this side where we are today, because we still have to develop new antibiotics. We don't know what the long-term impact will be with some of these new drugs. So we're still talking about, and the foundation is still focused on developing new daily therapies to treat the symptoms of cystic fibrosis. Now, I don't have the time, and you probably don't have the patience to go over all that we are doing with respect to this, but this is our pipeline. This is the pipeline of new therapies. You can't read it. That's intentional, because I want you to go to the website. And we have a very proactive and interactive website now that really goes over each of the drugs that are being developed. And some of these drugs, we're talking about new antimicrobials. We're talking about nutritional. We just had a really very, very exciting meeting with transplant surgeons and so forth. We got a new consortium for transplant working in the CF Foundation. We're looking at anti-inflammatories and some of the mucociliary clearance. So the reason that I show this, and I really intentionally wanted to make sure you couldn't read it, is the fact that we have, I've got to go in the right direction. Go to the website, go to cff.org, go to the interactive website, and if you punch on ivacaftor or lumacaftor, you see where the clinical trials are being done, what clinical trials are underway. You put in your zip code, you can find out where's the local one to go to, and everything. So that's really, really important. But once the going away message is we are still working to develop new anti-infectives, anti-inflammatory, nutritional components, and so forth. We have not forgotten about that, okay? But again, that's a daily therapy. And going back to this slide that I showed here, a PowerPoint, that's a slide, right? That ages me too, like encyclopedia. Okay? Treating the symptoms of cystic fibrosis to make treating the underlying. And those are called potentiators and correctors and all of those kinds of things that we are looking at in terms of opening up that channel so that chloride could exit. But those are daily therapies. You have to take those once a day, or several times a day, several times a day. And, you know, we're seeing some improvement in lung function, some improvement. You know, obviously, I think there was about an absolute about 8 to 10% with Kalydeco in the G551D patients. It was about 4 to 5% absolute improvement in lung function with Orkambi in the Delta patients. But wouldn't we like it to be more than that? To be better than that? And that's where so much excitement is now going on. For instance, Vertex, if you go back to looking at go to the website, has a 3-drug cocktail that the Foundation has paid for. It's not only Kalydeco, it's not only Orkambi, which has both Kalydeco and lumacaftor in it, and it's added another corrector that we paid for. We put another $75 million. That drug is going into human trials We've already done some phase 1, but it's going into human trials before the end of the year. From what we see in the test tube, that 3-drug combination is even going to be better. It may be double the impact, or maybe more than the fact of Orkambi alone in the Delta 508 patients. It also might work in those patients that have one copy of Delta 508, where the heterozygotes, where they do not have the other copy of 508, but another mutation. Because Orkambi doesn't work in those patients. Orkambi is not enough for, well, we don't know yet. Still the trial is going on. But certain of them that is not enough for those patients. But maybe the 3-drug combination is going to be better. And the other important thing is, and I'll show you my final slide in a couple minutes, is the fact that we have many drug companies like Pfizer, in addition to Vertex, like Novartis, like Genzyme, like AbbVie, and other companies that are trying to make better small molecules. Better ways to open up that chloride channel, all based upon good scientific opportunities and scientific method, and all working in the right direction. The better and the more chloride ions we can get through that door in a finite period of time, the better off the patient's going to be. And that is really the goal of all of that. So, but let's talk about that. So these are still daily therapies. Daily therapies. What if we could move back to maybe better approaches, maybe get even more production, but maybe they'd be monthly or every 6 weeks? Wouldn't that be better? The burden, even though they are pills, but still, just remembering to take the pills. Mimi's smiling up here because I am not a very compliant patient. Yeah, I look like he's making a face. I miss my midday one all the time. So, but anyway, let's move back to there's a clinical trial going on now. Now, remember, and ask the kids in junior high, probably elementary school, DNA makes message, makes protein. That's the triad of molecular biology. If you have a mutation in the DNA, you're going to make bad messenger, you're going to make bad protein. So what they're trying to do, we're correcting the protein now. What if we could correct the message and put that into the cells? And read through those mutations and get past them so that they are, when it's transcribed, it would be normal protein. Not even talking about defective protein, we're trying to get normal protein that's produced. There's literally a clinical trial that's going on right now called ProQR, where they're putting messenger RNA into patients, little segments of messenger that can jump over and read through and produce normal CFTR. That's a clinical trial. We don't have the results. I think the results are going to be presented at the North American meeting or thereabout. But that was a company that was founded by a father whose child had cystic fibrosis. And he went out and got Henry Tremere and other investors. They got $200 million. We provided them the seed money, early seed money, and then they got $200 million to explore this. But that could be a monthly therapy. It could be better than what we know about already. And then, so we've gone from daily therapies to treat the symptoms, daily therapies to treat the potentially basic defect. This is basic defect with the messenger RNA. But what about, could we really go back and think about one-time cures? One-time cures for cystic fibrosis. And the technology exists today or is beginning to evolve that will allow us to think about that. For instance, you and I, when we produce cells and cells go off the skin and so forth, cells are constantly evolving from what they call stem cells. We all evolved from, one cell, 2 cells, 1 cell, I guess. 1 cell, and then we've evolved to where we are today. In the airways, there are probably, what's the number that they say? It's 16 different kinds of cells in the airways, I think, that are, they got some with cilia on it, some produce mucus, some have globules in it, and so forth. But they all evolve from so-called stem cells that are built in the submucosas, in the cells. And these cells continue to differentiate all the time. What if we could get those stem cells out of the airways, put them in a test tube, and correct the mutation, put them back into the airways, and every cell from the progeny of those cells would be normal, correct? Would be normal. That's what you would want. That technology is beginning to evolve. And the foundation has committed over $30 million to bring that effort forward. Now, that's not going to be here 5 years from now. That's not going to be here 10 years from now, most likely. Don't forget, it took us 12 years to go from our first collaboration with Aurora till we got our first drug. But you got to make the investments today. And this is all talking about stem cell biology. Can we identify the stem cells? Thinking about, and now there's a new technology called, and I'm not even going to show a PowerPoint, I'll just really confuse you, called CRISPR-Cas technologies that allow them to be able to put a little piece of DNA in, it chops up the gene where you want it, and it inserts a normal copy. If you want to read about it, it's probably mentioned once a week in the Wall Street Journal and so forth, because it is the cutting edge of molecular biology today. So we are investing in that. That's what the future holds for us. And fortunately, our foundation is making the investments today. NIH is making the investments today. Howard Hughes is making the investments today. And going to the next slide. Right time out? Oh, no. Yeah, this is good. Hey, I'm finally got to go in the right direction. But this is why we need to be so excited about the future. It's our community. It's the fact that we got the great caregivers. But these people, these groups, these companies are all looking at treating the basic defect in CF. We've created a competitive environment. People talk about the fact that, and it is a reality that, Kalydeco or Comi are expensive. But because they are expensive, it's created a big market. And everybody wants to participate in that market. And our CF Foundation has supported many of these different groups to get them involved in cystic fibrosis. $56 million to Pfizer. And I looked at all of those. And the other thing that I think that last slide, next to last slide probably, is the fact that we have just recently we've expanded our own discovery laboratory in Lexington, Massachusetts, to where we have now 20 scientists that are looking at finding new molecules, working with industry. Why should industry set up an assay when they could go down to the street to our own laboratory and do it, in 2 days instead of 2 months or 6 months and so forth? And so we started this a number of years ago. It's grown to be a great resource, and we just had the rededication of it a couple of weeks ago. And to show you what this group is doing, they're doing things like gene editing, sequencing, theratyping, cell bank, but they're also looking at, as I said, PTC read-through. This is looking for screening for the X mutations. I'm sure there are people in here whose children or themselves have the X mutation. That's just a tougher one than I've talked about, because no CFTR is produced. It's very difficult to get. And so they're screening molecules to see if we can get that. And that's probably going to be one of our hardest problems. So that's being done in our Boston laboratory. They're also looking at gene editing and things like that. So I hope through this presentation that I have given you the kind of optimism that I feel that exist for us to continue to impact on the patient with cystic fibrosis. You know, I talk a lot about the drugs. I certainly don't have time to talk about the other things that the foundation is doing. Obviously, we got the mental health because I love Marlene's friendship. We got the mental health. We got the mental health issue talked about here, and but the foundation is doing more for adults. We have our hotline to help patients get Social Security. We have lots to access. So it's not only thinking about adding tomorrows, it's talking about the foundation's ability to live today. So again, Jim is here from the foundation. Jim, where are you? From the foundation. Jim, you want to make sure you pick these people's pockets before they leave? Okay, I'll get them signed up to Great Strides. Anyway, thank you all very, very much. I hope you go away from here this morning feeling more optimistic. We are in a good place, but I can guarantee you we are well positioned to be in a better place tomorrow. So thank you all very much. Do we have time for questions?